So yeah, thanks a lot for coming. I appreciate that. So this talk is about what I would like to call the quiet revolution of high-throughput experimentation, the change of culture in chemistry, and how it will change drug discovery forever. I think at least a large portion of you is quite confused by this title, and I think that's the point of this talk, that this is not really talked about, I think, that much. And the main goal for the talk is to first familiarize you with the topic, like maybe you can benefit from this revolution, from this change of how we do chemistry. But second, encourage to contribute to that as a researchers, because we need, I think, more people working on that. So first of all, quick, let's say, intro to organic chemistry. So if you know Breaking Bad, you have a very good picture of how chemistry looks. If you don't, no worries, I will just talk about it. So no worries if you don't know this culture reference. So Breaking Bad was about Walter White, who was a brilliant chemist, who invented a very innovative way to make a quite illegal molecule in the context of that series. And what was really well pictured is that there is this aspect of human ingenuity in coming up with how to make molecules, and also the series was really about the impact, financial impact of that innovation. That's also very important. And also, because that was about, that illegal molecule was a drug for those who haven't watched the series. And also importantly, throughout the series, he was making that molecule very manually. And very surprisingly, that's how often chemistry looks like. We outsource chemistry to China and other countries where labor is cheap. There is very little happening in Europe and the US. So I think this series is actually a very good portrayal. Now, I'm not sure how long is this talk, but I think it's shorter than 1.5 hours. So I would really welcome questions and stuff. And also, even if we run a bit longer, I can just cut some of the material at the end. So anyway, I really encourage questions. So you have seen this slide pretty sure many times, but I wanted to point out one thing about drug discovery pipeline that will be important for the next slide. So what is important is that per one approved molecule that goes to the market and you can buy it in pharmacy, say, around 9 others have to fail. And that's very important because that means that we have to actually repeat the earlier stages multiple times. It's not that you have to go, for example, through preclinical trials once. You have to go through them 10 times, at least the phase 1, if you want one approved drug. Of course, that's a rough estimate, right? This number evolves. So chemistry, try to think about what percentage of cost of a single molecule that becomes a drug is chemistry. I ran through this exercise, and I was quite surprised by the outcome. So this is based of a paper, How to Improve R&D Productivity. And you have to do some extra assumptions, but the number comes up to 36%. There are several reasons why it's so large. So this is the yellow part. The other parts are clinical ops, discovery biology, and other fixed costs, for example, related to regulation of the process. Now, why is it so large? One mental framework that might be useful is that we have to repeat chemistry, the discovery part, multiple times, at least 10 times to get one molecule, because clinical trials just fail so often. Second, interestingly, there is manufacturing as well, which is often forgotten. So when you go to clinical trials, phase 1, phase 2, phase 3, this is the moment you have to manufacture your drug at scale. It has to have high purity, it has to have understood properties, and it has to have many other properties, and that really drives up the cost. So like 1/3 or like up to 40%, close to 40% of cost is chemistry. While I think it's fair to say that when we talk about drug discovery in the news, let's say, but also in research, we almost always emphasize very strongly the biological difficulties, but not as much the cost of chemistry. And something that's hidden by this slide, but I will talk about this later, is that also chemistry is a big factor contributing to the fact that drug discovery process takes so long. That's on the side of costs, but I think a more exciting aspect of why chemistry is important, that breakthroughs in chemistry, kind of related to Walter White and Breaking Bad, breakthroughs in chemistry lead to novel drugs. The picture I have in mind is the space of all potential drug-like molecules. This space is huge, by many estimates more than atoms in the universe because of the combinatorial nature of chemistry. It's put between 10 to the power of 20 to a ridiculous number of 10 to the power of 63. But then on this very planet, we made like 1 billion, maybe, molecules in terms of like tested things for drug discovery process specifically. One of them, or a few of them are available on the market, like aspirin here shown as a graph. But I think what's most interesting about all of this is that there is dark matter of the molecular space. So a large subset of space that is never approached by drug discovery processes because making those molecules is just too expensive. If a molecule, at least its early stage before lead optimization, is to enter the hit discovery phase, it has to be available for around $300 per 10 milligrams. Otherwise, most big pharma, most biotechs just won't buy those molecules and test them biologically. So the limits of chemistry are the limits of our drug discovery. Something very, to paraphrase a known saying, something often not appreciated. So to illustrate that with a specific example, here is a Nobel-winning reaction Suzuki coupling. So it is between an aromatic halide, in this case, and an aromatic boronic acid, shown here. And it produces, basically the way to think about this is that it fuses two rings. So if you don't know much about chemistry and molecules, there are two rings, aromatic rings. They are flat physically, kind of, and they are fused. So you get this, a bit of a pancake molecule, if you will, if you were to think about it from the perspective of its shape. And importantly, that bond between an aromatic ring and aromatic ring before Suzuki coupling was basically impossible or very difficult to do. So in 2010, a Nobel Prize in Chemistry was awarded to people inventing this chemistry, in particular Akira Suzuki, who is still alive. He's the oldest of the three. But I mentioned that Suzuki coupling gives you compounds that are flat. And you can actually see, I think, reason about how Suzuki coupling has changed the shape of medicines or similar chemistry. Of course, it's hard to talk about correlation versus causation, so caveat, but many agree in the field that there is this trend. So here's a plot based on a paper from 2021 where properties of medicines were plotted, were shown versus time of the discovery of a given medicine. So 3 properties. sp3, which is the percentage of carbons which have 3D neighborhood. So sp3 is from hybridization, but if you were to think about this visually, on the left here we have, do I have a pointer? Here, okay, thank you. Sorry? Włączyć. Oh, okay. Good, thank you. Yeah, so this is sp2 carbon, which is a flat part of the molecule, and here is sp3, so it is like has tetra shape. So basically, the percentage of such atoms will track how 3D complex is a molecule, if you will. Now we'll also look at the median number of aromatic rings, and also we'll look at how many of drugs have at least one heteroaromatic ring, which again is something much easier to do with Suzuki coupling. And if we look at statistics, let's call them old drugs, so things invented until '89, have, let's read from the plot, the median percentage of sp3 atoms is like at 40%, and they have like one like 20% of them have a heteroaromatic ring, and they have on average like one ring. And you can quickly see the trends So there is a huge growth in like the like ring complexity of molecules, but interestingly, there is a decrease in complexity in terms of 3D shape. So our medicines that we ingest, let's say, become more flat over time. I'm not saying that's bad for sure, like we don't know if that means that those molecules are less potent, but it is an interesting trend. And you could overlay that with the prevalence of Suzuki coupling. So, of course, it's hard to estimate that precisely because, well, you have to make some assumptions. But let's say 10% of reactions used in synthesis of late-stage molecules can be estimated to use Suzuki coupling. Again, you can estimate it in different ways, but always the trend comes up that there is more Suzuki couplings with time. So you have this trend, it's a correlation, more Suzuki, less complex in terms of 3D shaped molecules. But I personally think there is a causation. So an example of both are anti-cancer drugs, an example of an older drug and a more, let's say, recent. And the one on the right is an example of a molecule I would call more pancake or more Suzuki coupling rich, maybe. And if you were to plot their 3D shape, you can see that as well, that this is just complex and this has like quite a lot of planar shape to it. Again, the thesis I'm making is that the invention of Suzuki has increased the number of those on the right and versus those on the left. And again, like the limits of our chemistry define and bias the limits of drug discovery. Chemistry is a tool of drug discovery, but one that influences us a lot. Now, one perspective I like on where chemistry is going is from this paper, The Importance of Synthetic Chemistry in the Pharmaceutical Industry. If you would like to a more, let's say, complete story of chemistry versus drug discovery, it's a really nice review. I recommend it. Is that now we are in this phase of the evolution where, based on the medicinal toolbox we have discovered, so Suzuki coupling and many other reactions that we have just recently, maybe not recently, but in the last 50 years have discovered, we are really changing the way we are doing chemistry. And two big trends are using, let's say, biology for chemistry, so in particular using enzymes. This is something I will talk less in this talk, or like won't talk about it. And high-throughput experimentation, where we put more emphasis on experiments and seeing what works. That will be the stock. And the dream that all of us have in the field is that in the next, I don't know how many years, I don't know how long it will take, but you will be able to basically have any molecule you want as long as it is physically sensible. You can think a bit about like there was this machine in Star Trek where you could like basically have anything. That's where we want to go, and I think, I think we'll get there at some point. And for those of you who are doing de novo drug discovery, I think always you have to include right now some form of penalty on synthetic complexity. So when your model generates, for example, GFlowNet, RNN, spits out new molecules, you have to trim down the creativity of the model. So in a way, you are trading off potential potency for what can be synthesized. But what we are hoping for is that, is that with some, with some effort of the field, we will be able to make any molecule instead of, adding all of these penalties and things like that. Cool. Yeah. So that was kind of what is organic chemistry. Now a bit about what is the closest obstacle that I think we can solve as a field that will push us towards this agenda of molecular editing and other promises of chemistry for drug discovery. And by the way, again, if you have any questions, just raise your hand during the talk. I think we have plenty of time. And after this section, there will be like a small recap, so that also will be a good opportunity. Okay, so sorry to emphasize this point, but I think it's really important. One drug approved is like $600 million in capitalized, to be very specific, cost in chemistry alone. And that's an important problem, I think. The breakdown of that is that it's half discovery, half manufacture. And one point more why this is important. We have really great directions for cutting down other parts of the cost. I think there is great research in algorithmic methods. I think there is a lot of deregulation. I think there is promise of better models of human biology, things like organoids and other things some of you might be familiar with. But somehow the story on the chemistry sometimes is a bit more bleak. Like we don't have that many maybe very pronounced trends that will for sure drive down dramatically this cost. So that's another reason, because in 10 years, if we don't advance a lot chemistry, you know, we might have a big, huge bottleneck that we haven't thought about earlier. So why is chemistry so expensive? First order approximation of that is that chemistry is very intensive, labor intensive, just like in the Breaking Bad series. So of course there has been a lot of push to build self-driving laboratories, right? So basically robots doing the chemistry. We have robots everywhere in factories. So why not just have a molecular factory that spits out molecules, problem solved? And indeed, if you go to many companies, you will find very, very advanced demos, videos of like just this working, like beautiful robots moving things and basically doing the dream of human not making molecules but the machine. But unfortunately, all of these are advertisement to a large degree. So it's really difficult right now to have economically sensible or like really drive forward drug discovery with automation chemistry. It's easy to do demos, to be honest, but it's very difficult to do something that really revolutionizes chemistry because potentially the assumptions are wrong to an extent, and this will be the last section. So what is the reason, if you trust my judgment that there is no yet large-scale automation chemistry, what is the reason? I think it's in the name of the field. So what chemists like to say is chem is try. So data, both from Molecule 1 and papers that are disclosed, clearly shows that chemistry is just very hard to predict. So it's a very unpredictable domain, you could say. So what do I mean by that? How you can judge a failure or success in chemistry is that you have a well-intentioned, let's say, PhD-level chemist going into laboratory and trying to make a reaction. Let's say try to run Suzuki coupling. And then you can say whether it has worked or not. A good one more analogy about chemistry that might be helpful is cooking. So chemistry is a lot about mixing stuff, boiling stuff, just like cooking. So in particular, if you were to make a tea with cold water and stop after 1 minute, that would largely be a synthesis failure in the sense you wouldn't get a tea. So how often do chemists fail to achieve desired product? So one paper by Merck showed that 35% of backward Hardwick couplings, which is another coupling, have failed. And that's a lot, because if you want in a robotic platform to chain together 3 such transformations, you quickly get to like below 50% success rate. And imagine your self-driving car having 50% of chance of basically running into something, right? Another paper showed 32% failure rate of Suzuki couplings and ABV. Defined slightly differently, so here after purification, which is a common step in chemistry workflows. Oh, sorry for that. At Molecule 1, we kind of see similar things. So we do some synthesis completely designed by humans, and we have like 40% failure rate then, around. And we also generated scale reaction data where we do random Suzuki couplings, not defined by anyone, but just for training models. And like by some measures, like 70% of them do not succeed. So to help you think about why automated laboratories do not yet work, which is the kind of problem, is that they do not work for the same reason that self-driving cars do not yet exist. I think they will, but not yet. So self-driving cars are not bottlenecked by robotic advances. I think the car is really good, but by the lack of the brain. And the field has made amazing progress in building the automation of chemistry, but I think what is lacking a bit is progress on the brain of chemistry. In particular, if humans have very high failure rates, meaning that if humans design a synthesis in the laboratory and it fails, then you could argue that if we use humans to script laboratories versus learn from data, then those laboratories also will often fail to produce output. Maybe one more axis along which synthesis lack of automation is problematic. So yes, we can outsource things to China where labor is cheaper, or India, but that is problematic. So the statistic is that roughly 70% of big pharma outsources at least partially synthesis to China, and that relatively little synthesis is done in Europe and the US. And why is that a problem? Well, you could make different arguments, but one that I think is very powerful is that right now there is a lot of IP leakage. So big pharma sends their IP, molecules that are bioactive, to China, and there are very good arguments that this IP is lost there, and there is a lot of espionage. So that's also a geopolitical argument. I hope not a controversial one, given a lot of that is now quite well documented. So there's also a reason we need to have automation, because labor in the Europe and US is just too expensive to satisfy the needs of drug discovery. Okay, so that's the problem. And for a significant portion of the rest of the talk, I will talk a bit more, let's say, concretely about what Molecule One does, because that's where I feel I can talk about something concrete. Molecule One really started from, like, a lot of it started from this perspective that I wanted to kind of open with. So let's say this is a difficult problem. Chemistry is a difficult problem. Like, it's still very unpredictable, but at its essence, it's a prediction problem. So you predict how to make a molecule. You plan how to make a molecule. So here is something that Ilya Sutskever, a pioneer of deep learning, has said a while ago. He had this slide. So if you have a hard problem, if you have a lot of good and labeled data, and you train a big neural network, success is the only possible outcome. It is very simplistic, but I think, especially given that this was, I think it was very prescient because it came before all of LLMs and stuff. But I think there is a lot to that. Deep learning is a machine that can learn from data used for interpolations. So we can shift the challenge or the problem more to data generation and try to, in that way, to solve chemistry. And that's a new way to solve chemistry because, again, high-throughput experimentation hasn't been a big topic in chemistry until last decade or so, to simplify a bit. And what I think is even more exciting is that you not only make chemistry predictable, you hopefully discover novel chemistry that I think we could call precision chemistry. So you discover new ways of making molecules that are very amenable to automation, that you can predict outcomes for, that are very robust and very reliable. Through different things. For example, you can invent a new catalyst, you can invent a new procedure, or you can invent a completely new reaction class. And that type of discovery, in general, is very important, not only for making chemistry predictable, but also solving many other challenges in material chemistry as well, such as carbon capture. So that's all from the intro. Maybe I will summarize briefly, and maybe you will have some questions. And if not, we'll go to, let's say, a bit more technical part, to an extent. So, yes, please.
On the previous slide, I just want to sort of better understand this. Are you saying that out of the known chemistry, when you say predictable here, do you mean currently predictable or predictable sort of in general, what we can hope to predict ever?
Right. Okay, so maybe a concrete example. So, for example, there is a reaction class backward coupling, which is very useful, but small changes of temperature lead to different outcome. So in that sense, it is hard to predict outcome for, because you need to be very accurate and precise about temperature prediction. So the idea is that maybe some new reaction classes are just as powerful, but much more robust. So it's about both discovering novel chemistry, but one that is also easier to run by humans and machines, I hope. So in terms of Venn diagram, some of that is intersects with known chemistry, but many of those will be completely novel and unknown to us. And hopefully at some point, there will be multiple Nobel Prizes for new reaction classes that are in this kind of category. And of course, many are of this type. Like there are many reaction classes that are known to be very robust. For those of you with chemistry background, there is click reaction, click chemistry, which is very robust. It just works, but has other issues. So a small recap of what I wanted to say. I made a claim that organic chemistry is the beating heart of drug discovery, with 1/3 of costs, but also the shape of molecules that we have approved is fundamentally influenced by chemistry. And we have to make chemistry cheaper and better to make medicines better and faster, right? Like I think it's both better medicine, but also make them faster. And the core challenge and obstacle that we are here, I think, tackling, one that I think can be tackled, is predictability of chemistry. So the issue that outcomes of chemical reactions cannot be predicted. You can predict whether a tea will form after 1 minute if it's very cold water, but you cannot, and humans cannot, even one trained for years, predict well outcomes of many reaction classes. And the solution is high-throughput experimentation, which means generating a lot of data about chemistry to feed models. So that was the intro. Do you have any questions before I go further?
Yeah, thank you for a great intro. And I have a few questions. And first of all, I also actually was following this RoboLabs and was excited first. And it's been already maybe more than 10 years that they were once in a year, they are appearing again and again. So how do you think, what are the main, I would say, stopping points, why it's not so popular, why it's not used? Because in the image or on the video, promotion video, it's great, like it's really beautiful, everything is working, but why we're not having them still?
You mean robots in synthesis?
Yes, exactly,
right.
Like fully automated labs that you were talking about.
So I think there are factors. I think a very important factor is unpredictability of chemistry. So those laboratories are often designed in a way that they can kind of do a small diversity of molecules because a lot has to be controlled for all of that to work with 100% reliability. So it's basically like self-driving cars, to maybe repeat some points, self-driving cars not being yet on our roads in Kraków, not because the robots in the car are not good enough, but we kind of do not have the visual intelligence to 100% reliably drive the car. So that's how I think about this, but there are other factors as well.
And what also do you think about, because probably all the chemical practitioners know that setting up a reaction is maybe 20% of the effort,
yeah.
And all the other effort most of the time is purification and isolation, identification.
Yeah.
How do you think we are on this right now on these areas? What are have we solved it by these robotized labs, and are there any approaches automatized for these kind of steps?
Yeah, so there is a section kind of dedicated to that. In short, there are two aspects of that. One is just do not purify compounds actually, and you can still do a lot of drug discovery, and we'll talk about this. But second is that as you generate this high-throughput experimentation data, you also generate data often about purification because you run LCMS analysis, and you can train that on that as well. There's much less research on that, but I think it's can be thought about again as data bottleneck problem. And then if you predict well purification in the sense of what method to use, then robotizing that is not that difficult. The problem is that we try to purify compounds and the method we try does not work. Like that's the, I think, core problem. So I think again it's kind of a prediction problem.
Okay, thank you.
Thank you for the questions.
Anything else? Well.
So as much as I understand correctly, so one of the solutions is actually to make it more predictable so that we decrease the number of, like the percentage of failure experiments. is it like the only solution? Like, for instance, I can see like one very intuitive one. So we decrease the, we stay with the failure rate, but we know how to respond to that. So like, we know when it failed.
Yeah, I think that's of course both ends, and you're absolutely correct. There is end of predict what works and end of being flexible. I think both should be attempted. The being flexible is just very difficult because many things can go wrong in the lab. Like things to blow up in the lab. Like things really can go in any direction in the lab. So you have a lot of things to respond to. That's the problem with self-driving cars as well, by the way, because they cannot drive on predictable roads like we can in chemistry, right? So they can have people do really weird stuff with self-driving cars. I don't know, they walk dressed as cones to fool them, you know? So in chemistry, we have this easier component that we can focus on reactions that are more easier to predict. Thank you. Any other thoughts, questions? Yes.
Hi. Just to follow up on that slide that you had previously with the two bubbles of known, yes, and predictable chemistry. So maybe I'm misunderstanding, or maybe you will cover it later on, but are you sort of claiming that we can discover new chemistry just through experimenting?
Yes.
But is there a way to structure the space in a way where like searching through this and learning is actually feasible rather than just like random?
Oh yeah, of course. I think the core thing is to have high throughput platform, but then you can use it in smarter ways. But my claim is that without high throughput platform and continuing what we do, kind of one experiment by another, kind of, we are not fast enough. Like that's kind of the claim. But it's not saying that, you know, search algorithms do not matter. They matter a lot.
I see. But do you think that the ratio of like new reactions to the space is feasible, where you can actually, like this is a tackleable problem through just high throughput?
Perhaps you need advances in both. My claim is that you cannot do without advances in high throughput, but maybe you also need to add on top of that like AI scientist type of algorithms. And for sure, we're also exploring things like that. So yeah, not a dismissal of being smart in search at all. Any other questions? Well, thank you for that. The more questions, the better. So a short introduction how to think about molecule one, and then I will talk about some technical results. So the company was founded in 2018, and the way we like to think about us, we are just solving this problem that, let's say in aggregate, 50% of reactions fail, and this limits our ability to make progress in drug discovery. So we started in 2018 by, I think, having first broadly available commercial deep learning solution for synthesis planning. So the task is kind of something that Walter White could do from Breaking Bad. So you have a molecule and you design how to make it as a sequence of steps. We have a talk by Marvin later. I'm not sure if Marvin, you will talk about synthesis planning, but this is largely based on foundational work by Marvin from around 2017. So it takes human to design a good synthesis pathway hours, while such a software can do it in minutes. We have partnered in 22 with CAS, which is an organization from the US that has the largest data set on reactions to feed the platform. And this year also, kind of as a part of this journey of the software leg of the business, we have won a retrosynthesis challenge, retrosynthesis software challenge, where the goal was to propose best synthesis planning solution with focus on manufacturing, part of the drug discovery process. And since the beginning, our focus was on training deep models that do not hallucinate. Before it was even a word, actually. We used generative AI in 2018, and they were just as hallucinating as today. So interestingly enough, we actually used similar techniques back then that are now quite common. There's a coincidence. So that's one part of the business. The way we evaluate this software, so I will be starting to get into a bit more technical stuff. The way we evaluate this software is largely, though not only, asking PhD level chemists to tell us whether the pathway is correct. Interestingly, almost no research does that. So most research evaluates pathways in other less direct measures. Of course, the most direct would be experimentation, but before that, you can ask an expert chemist what he or she thinks about the pathway. So each block here, like that's the number of targets for which we run evaluation. Greens are okay, reds are not okay, and no pathway. So the system said, no, I cannot make it, is gray. And here's comparison to two solutions. I could include more, but the bottom line is that the system is accurate and like it returns like 80%, depending on the target, 80% of pathways are just okay for PhD level chemists. And I think that's important because this really frees these hours of work. And in particular, when a chemist has to evaluate hundreds of targets, that's a really good screening solution. And that's how it is used right now commercially. But if we want to really solve chemistry, we quickly notice that the data we use for this software is just not good enough. So this software and similar softwares are trained on reactions disclosed in papers and patents. And as we all know, as you all know, we publish only successes. Who would publish a failure, right? So we have only basically greens, only positive reactions in the data set. So how do you train a model which predicts failures if you have just successes? There are techniques for that, but it's very difficult and it doesn't really work. Also, importantly, here is like different conditions. The issue is that we have these reactions disclosed, but no one is testing multiple sets of conditions per reaction. So the reactions are not comparable in terms of influence of conditions. So it's also really difficult to understand how, let's say, a temperature would improve outcome of a reaction or make it a failure. That's really difficult. So the data is not there. And we were a software company. So, you know, it's like we in the executive team had a software company, had this problem, we were like, what do we do, right? And we sat there and we're like, I guess we have to open a laboratory. And that was really scary. Maybe for some of you who work more at the bench, it's less scary, less relatable, but for people working on the computer, opening like a physical lab was a leap of faith. So where we are now, since 2020, we have been generating vast amount of reaction data. So each reaction point, each data point is outcome of a chemical reaction. Here on the right is our head of chemistry standing to those, let's say, data points. So these are 384 well plates. So 384 data points in one plate like that. This has been actually, as far as I know, the largest reaction screen of this kind. So we have run 60,000 reactions in matters of 3 weeks. We have been focusing on 4 reaction classes at the moment, and then growing beyond that this year. And in total have generated 230,000 reactions and counting, really going far in this direction of we can solve this problem with data. And this, as far as I know, is the largest data set of this kind that is at least publicly announced. Maybe there are some more less public. So we have really tried to execute hard against this belief that you solve hard problems with data and deep learning. On a plot, that's also a fun way to visualize it, though focus on 3 of those reaction classes. You can see the progress of the laboratory since 2020. And there is this like fun part of our journey. I'll talk a bit about this when we had a good incentive to run a lot of reactions very quickly. Yeah. But then in the life of any company comes a moment, how do we make a lot of money? And like that was fun, but monetization of high throughput experimentation is difficult. You have a data set, but it's not easy to sell this data set. I won't go into details why, but it is a challenging proposition. So how do you make a progress business-wise? So the way we started to operationalize that is that we can commercialize discoveries. So here on the right is a t-SNE plot, so an embedding of molecules. Each dot is a molecule from our space of molecules that we now offer for synthesis. So we offer physical samples of what we discovered how to make. Kind of like in Breaking Bad, but our molecules are fully legal, just to be clear. Though we did get some questions about that, but we didn't answer them. I can talk about this after the talk. So they are colored by similarity to the most popular right now molecular space called real. And basically most are blue, which means they do not have a close analog. in the real space. So we offer differentiated molecules. So if you do drug discovery, we offer the promise that for with cheap molecules, you can go beyond what you can get from other spaces. So it's a new axis, right? Like you can take a de novo model like GFlowNet, but look at novel parts of the chemical space with our space. That's the promise. As far as I know, it's the first space of molecules based on such high throughput experimentation data commercially available. At least, or maybe some other is like concurrent, but this is pretty new. So basically the flow is chemical knowledge to commercialization. And in summary, our journey is from software to data generation to monetizing discoveries, monetizing our knowledge about chemistry. That's the story of the company. So I wanted to talk a bit about that screen, right? So that's that reaction screen. And basically try to think together what you can get from 100,000 chemical reactions in a given reaction class. Again, that's a lot of data point in our domain. In biology, that wouldn't be that many actually, but in chemistry, that's very rare to train on such a data set dedicated to one reaction class, especially if it is not mined from like literature or patents. So yeah, I wanted to say that this data largely was generated, but it has been being generated since the beginning of 23 actually, or mid 23. So let's come back to the commercial product. We have to make money. So Space M1 is a space of molecules that someone can order. It's like a catalog, a shopping list, if you will. So you can imagine, again, the whole space of molecules, all of them. It's huge, even like 5 billion compounds if you consider just Suzuki coupling products between any building blocks you can buy. Then we need robust AI that selects from that a subset where we are certain we can make those. And ideally, this subset will be differentiated from what others offer. And the workflow of the product is that someone comes with compounds they want from our catalog, maybe discovered by de novo models, and we ship them as a physical sample. Again, just to make sure this is clear, it's not an API. It's it can be as API, but it is a physical product that gets shipped back to the user. So again, a very kind of physical problem. So I think most of us here are computer scientists, so we can start to think about this as a prediction problem, or like AI problem. Maybe most of us are AI engineers, right? Scientists. So if you want to build this product, what do you need? You need a model that can select highly successful subset of molecules and can generalize very robustly. And by the way, this generalization in robust way, that's the Achilles. That's the weak point of AI right now. You know, if you take even the best AI and apply them in very novel situations, that's where they start to break. And we actually need to solve that if we want to select from this virtual space of all molecules. So we need very robust models that work on very high diversity of potential molecules. So the whole data generation workflow is that we buy building blocks from providers. So building blocks are molecules you react. Then we generate this large data set with semi-automated workflows, with pipetting machines, with autosamplers to analyze the reactions, and then we train robust AI, hopefully robust AI on that. So we frame reaction prediction for simplicity as classification. So let's say you have this Suzuki coupling, and you ask, will this reaction achieve over 10% yield using SFOS? So that's a catalyst, 90 degrees of Celsius. One word that I haven't yet explained in this presentation is yield. So basically, you can think about this as what percentage of my substrate molecules got converted into the product molecule. You want this to be non-zero, because if that's zero, it means you mix stuff and nothing happened, or something else happened. You don't need this to be 100% because it's okay to mix a bit more to get how much you need. 10% is often used in medicinal chemistry. So how do we evaluate reaction classifiers? Again, out of distribution is important. So maybe first starting with desiderata. We want high success rate in the laboratory. We don't want our chemists to go into the laboratory and attempt reactions that do not work. We are okay to reject some reactions. So That's the problem a bit with ChatGPT. I would say that it kind of has to answer every question or tries to. But in our case, we are OK with just saying no. And that really can drive up precision. And that, by the way, I think is very important in any business application of AI, while it is a bit less visible in AI papers, that you can just drive up precision by drawing down recall. That's kind of obvious, but at the same time, very powerful. Sorry. And as a metric, because any technical talk should have a metric, we'll just compute success rate, because we want this to be high. Now we'll compute success rate at 50% recall. So we basically take some group of reactions, we select top 50% that we predict to be most likely, or humans, there will be a comparison with humans, and reject the other ones. And in terms of the baseline, so if you had a random classifier, that would be basically the percentage of successful reactions. So if you were to take reactions and take at random 50% of them, on average, you would get the average success rate. And if this metric is larger by 2x between different solutions, that means you reduce to increase 2x the number of successes. And this is a real economical value. It's actually also a very important process value because your client trusts you, right? Because you do not say you can make molecules and then not make them in the end. In terms of how to build the test set, again, something I think very important in technical presentations that's often not clearly described. So you can think about the data set as living on a grid. You mix reagent of type 1 with reagent of type 2, so aromatic halide and boronic acid, if this is Suzuki coupling. And then we evaluate mostly on this subset, which does not entrain on the black subset. So this subset are reactions where both substrates were not seen in training. That's important because this is how we will use those models in practice when we serve our clients. To briefly take a look at the data set, a big chunk of it is like 8 different sets of conditions tested for every substrate pair. So it's really like high throughput and exhaustive in that part of the data set. And on average, some reaction conditions actually were very unsuccessful. In that case, we picked conditions quite random. This was because of the engagement with the client. And some of them were very successful. So there is a difference in terms of productivity of a condition set. On y-axis is the percentage that's successful. Though if you look closer at this plot, you will notice that even the best one has like only 45%. So you need a classifier, because if you were to randomly do reactions, you would get 50% of successes in this case. We see a lot of novel scientific knowledge in this data, so things that probably on its own, if researched more deeply, could be turned into multiple papers about Suzuki coupling, so that was fun, at least to my knowledge. I'm not a chemist. So one thing you can do is like look at the success rate difference between sets of conditions, and on here we have different functional groups. For example, nitrile, which involves nitrogen atom, has a very large difference. So one set of conditions can make molecules with this group, and the other struggles. And those things cannot be easily derived, mostly from existing literature. As an example, for those of you with more chemistry knowledge, here is a Suzuki coupling involving a substructure called tetrazole. So basically, as far as I understand this part of the molecule, it had 4% yield in that case, but it's very rare. So I haven't found many examples in SciFinder, which is a repository of reactions, with tetrazole group. But maybe you want tetrazole group because this seems like really interesting from biological point of view, right? Like nitrogens can form bonds. That's the reason there are nitrogens in DNA. They form these like sticky interactions. So maybe you want those molecules, but maybe in other spaces you don't get them because Suzuki couplings are hard with tetrazoles. So that's the promise of the data. A bit about models, though, quickly. So we want high precision and high robustness. So what models do we have? As the baseline, we have a graph neural network. I suppose it was shown at some presentations, so I won't go into details, but in short, you can think about nodes and edges having embeddings, and those embeddings evolve, and then you read out from them. So we introduced a novel model in the context of this project. So we call that UniReact. The reasoning behind model was as follows. So in general, transformers should overcome at some point graph models due to being more expressive. We see that with convolutional neural network versus transformers also. But we care a lot about out-of-distribution generalization. So maybe we want a sweet spot where we have expressivity of transformer, but we build into it some inductive bias. So solution is a UniReact, that's how we call that. It's a custom graph transformer that is based on, I highly recommend this research, Unimol V2 mostly, also inspired by some earlier ideas from research done at Jagiellonian University, which involves in particular 3D embedding of molecules. So the structure at a glance of the network is as follows. I wanted to include in this talk some technical aspects. So we have two substrates that go through a unimol block, which if you know AlphaFold, it outputs something very similar. So you have pair representation and atom representation. Pair representation represents with embedding, so it's not like attention, every pair of atoms. And then this is concatenated and passed again through unimol block that is not pre-trained, and then there is a prediction head. So that's the architecture. We also use another trick, which I haven't seen that being used as much in the field, so that could be useful maybe to you. Of course, it is well known. It is known, but I haven't seen that much in chemistry and biology. So what we do is, because we need this robustness, we train an ensemble of models. So that part is very common, right? But each ensemble member is trained on a different subset of the data. So we call that a diverse ensemble. It has become an important part of how we do AI at the company, kind of in general. So in particular, you can remove like whole rows and columns of the data set. If you imagine the data set as grid of all reagents reacted with all reagents. And then you ensemble predictions. You could do later different things. You could maybe distill them. Maybe you could do something like more exploration where you add them dynamically. We just train and average predictions. So in summary, graph transformers, diverse ensembling, and also high prediction threshold. So we trade off precision for recall. That's the modeling at molecule one of this data in essence. Let me know if you have questions about that. Yes.
Yeah, thank you once again for the more technical part of the presentation. And regarding the first embedding of the molecules, I saw that you were using also a 3D
That's true.
kind of embedding. And because we all know that, well, I'm thinking about more from theoretical part, that it actually really matters what kind of conformer you're choosing for it.
Yeah.
And especially for this activation state, it really matters that sometimes these conformers are not the lowest ground state. Yeah. So how do you tackle this problem, and what kind of are there any trick to sort these conformers before?
That's a very good point. We haven't experimented with better embeddings. The kind of maybe weird thing is that Unimol actually corrects internally those conformers because it is trained with them, with those not necessarily the best conformers, to predict well-defined ground truth labels. So internally, as far as I understand, it internally tries to improve a bit this conformation, but potentially maybe ensembling different conformers would also help. So yeah, this is a very good comment that here something could be improved. Also, this architecture is probably too complex as well for this task. So I think we could potentially benefit from simplifying it.
Okay, thank you. And, sorry, another question, maybe a little bit more general, like for the future, because right now I see you have a really great and solid, like the synthesizable reactions, preparing compounds. And I know that like for a more integrated company, they also have like the part of predicting biological activity.
That's true. That's very important.
Like, and also working on the target identification, like, and all the things we were discovering previously in the conference. So do you have some plans on it, or you already started working on that, on this part of the task?
Yeah, so here, more to come, I think, but nothing we can yet talk publicly that much about. But yes, I think in general that's an important component. but also want to be uniquely a company that focuses on chemistry. We don't want to lose our DNA. So we are kind of thinking about that. Yeah, thank you.
Thank you.
So it's relatively well known that, with models, garbage in, garbage out. So when you have these experiments, you also need to, of course, be able to trust them.
Yeah.
So I was wondering if you sort of find some good way of balancing repetitions and doing more molecules, and whether you're able to use this sort of uncertainty from the data.
I think kind of one thing is ideas, another thing is execution. So I think a very important part of execution of such a product is a robust quality assurance. And we are continually improving in that. So it's about measuring, it's about culture as well. So yes, this is very important. And we basically measure how reproducible are experiments between days on the same day and try to keep this number as high as we can while maintaining throughput. So yeah, that's very important. Okay, if no more questions, then to the results. So how do you evaluate such a thing? Well, one thing is you can, again, compare to humans, but most importantly, of course, is just how many were successful. So on the held out test set, we, again, as the metric, have percentage of successes in the set of recommended reactions. So in that case, in this test set, 50%, or more precisely, 48 have succeeded, and that's the baseline that kind of we have to beat, let's say. Humans have improved over that. So we asked, I think, 3 PhD level synthetic chemists to predict whether or not a given reaction would be successful, of course, according to our protocol, and they got 60.8. Not enough for automation. That's too low success rate, especially if you were to run 3 of such reactions in a chain, you get 0.6 to the power of 3. Which of course drops very quickly to low values. The model got 87.1% in this metric, so that's over 1.5x increase over the baseline. And also starts to be at the level where we believe you can think about automation. Especially that you can potentially drive that higher by, again, increasing threshold. So maybe not recommending 50%, but 25. On another broader test set, because now we don't have to ask humans, we also evaluated different variants of the models I have just described. So graph model got how much? 55.6%. In that case, it's a different test set. So that's why this percentage is lower in general, because there we balanced positives and negatives to have a lower variance of the readout. Here we just run on the raw data set. Then diverse ensembling brought us 2% point gain. Then UniReact learned better from this data. I got to 65%, and then with diverse ensembling, we got to 69%. I think, so these 2 tricks, I think, on this data have proved to work, and we are exploring them in the company, and in general are enthusiastic about this. One thing that's kind of on the similar note as garbage in, garbage out, so do you need that many reactions? Like, why not run 1,000 versus 100,000? That seems extensive. So we computed here, let's say, maybe not scaling laws, but trends of performance versus data set size. So here you have training set size from 10K to 50K in that case. And here you have a performance measure that's precision recall under curve. So that's the results. Orange are UniReact, blue are the MPNN, which is a graph neural network. So interestingly, if you were to run 10,000 reactions, you don't see a difference between transformer-based model and graph model, which reflects what is happening as well in vision, where CNN versus transformers perform similarly at medium-sized range of data sets. And interestingly, you start to get reasonable increase in performance with data set size. With UniReact, well, the graph model has flattened. Is it universally true? No, it depends on the data set and specifics of the graph model. But what maybe can be an outcome of that for your implication is to give chance to transformer-based models. If your data set is larger, maybe they will work better, maybe not. I think it will depend on the data set. So that's about that campaign. Now a bit about the commercial product. It is a commercial product, but also I think very relevant for all of us here, for all of you here, because when you do de novo drug discovery, you have to constrain yourself to some molecular space. So it's basically about bringing more creativity to models, I would say. So again, we introduced space M1, which is a space of molecules that we ship as a physical product, to emphasize. And the workflow is that usually clients send us molecules they pick from our catalog, and we send them back physically. There are some variants of this workflow. And I have shown already this plot, so that the space is differentiated compared to real in our evaluations. By the way, to be transparent, there are other spaces we haven't compared in such apples to apples fashion, but real is the most popular one. Bottom line for us, though, of this plot maybe is less about commercial value, but more in the value of experimentation for discovering novel chemical matter. Because we kind of feel like we're just getting started, and we already kind of explored novel areas of chemistry. So we are really excited about how much we can push it by exploring a reaction class that is not in real at all. And we are now starting experimentation on a reaction class that no one is using in synthetic chemistry, literally, basically. So where can you go if you really push the boundaries of what you synthesize and the AI for chemistry? We completed some projects with clients. In general, we got consistent feedback. So one biotech said that chemistry already surpasses what we have been able to get from enamine and Wuxi. Another told us got good, if not better, what we get from industrial library synthesis molecules like molecules and enamine. Again, cases, I'm not claiming space M1 is better than those spaces. That would be not possible based on this data, but consistent with diversity. What we see is that our space has differentiated molecules, and we go beyond what is often available elsewhere, because we kind of take more risk, maybe, like we attempt molecules that look challenging, because we predict them with our AI to be not challenging. We also ran this experiment. So we have this feedback that there is good diversity. We're curious if we can find well-docking molecules. Before I go further, docking, to a large extent, is a random number generator. I am aware of that. However, it does tell you something about the space, and in many cases, you can expect to perform better than random with well-docking. So it's not that I think an in silico experiment can replace physical experiment, which I won't be showing, but I think with this caveat, this data is interesting. So we tested 7 proteins spanning different families, kinases, GPCRs, there is CYP. For each, we docked 1,000 compounds from real and from space M1. And as ligand activity, we docked using Autodock Vina, specifically docstring package for those of you who maybe are more familiar with this field. And ligand activity was evaluated using a metric called ligand efficiency metric, which is nothing more than docking score divided by the number of non-hydrogen atoms, which corrects for the fact that larger molecules just dock better in terms of the score. So it's a very simple experiment, I would say. On purpose, this is actually not more complex. You could do many things here. So in short, we saw that space M1 has many more, in that case, formally 10x more, highly docking, or like maybe not highly docking, but like active molecules according to ligand efficiency metric. So for example, for the kinase, so GAK2, which I understand stands for just another kinase, actually, which is a weird way to name your protein. It's 2.2% versus 0.17 in real. And importantly, again, we saw most of them to be novel with respect to real. Again, a comparison to real, I don't know how this comparison would work with other spaces. I think we still have to do that. So not to overclaim, but I think that's interesting. Again, I think it stems from the fact we have more of those, I don't know, like spicy molecules, if you will, right? Like molecules with interesting structures. Yes, please.
Just a clarification. What is Tanimoto? You used it before.
Oh, I'm sorry. Yeah, that's the jargon. So I'm sorry. Tanimoto is a similarity metric, actually in mathematics often called Jacquard similarity index. Basically, it measures how similar a molecule is to another molecule by first computing a binary vector of vector and then comparing to binary vectors. So large Tanimoto will mean that the embeddings overlap a lot, which corresponds to some degree with how chemists would say about similarity of molecules, but to a degree. Like that's a simplistic measure. But basically, if there is like 0.8, 0.9 Tanimoto, 1.0 will be max, then molecules usually are judged by chemists to be quite similar.
And that's, sorry, that's the similarity between the ones you find?
Oh yeah, okay.
And the subset of known solutions, or?
Okay, so when I say that those are novel with respect to real, or what I measure in this column, thanks for the question, is I take those molecules and look for the closest one in the whole catalog of compounds available from real. So that's 5 billion, no, now like more like 40 billion compounds, something like that. Yeah, sorry, that wasn't clear indeed, I'm sorry, and I should have introduced more carefully other molecular spaces, but yeah. Cool, so that concludes molecule 1 kind of part of the presentation. The last short section is about one particular way high-throughput experimentation can influence all of us, me included when we engage more in biology, by changing the way we test physical matter to make discover hits. And that is related to the question of purification. Before that, any question about, let's say, molecule 1 stuff? Yes, please.
Yeah, thank you. I wanted to ask about your comparison with the novelty of between real database space and your generated M1, because I was kind of was connected a little bit to the generation of the real space, and as far as I understood, you were mostly using quite, I would say, low amount of reactions to generate the M1. Yeah. And the building blocks, I also suppose, were coming partially from Inamine.
Yes.
So my question is how you achieved such degree of diversity and novelty, given that those reactions were already kind of encoded into the
that's
real
very
space.
Interesting.
Yeah.
So real, how they say, is constructed in this way that they very much narrow the set of building blocks to those that are very likely to work. So it's so real.
Experimentally tested, proven to be working.
Mostly. So the way real talks about how they construct, like I wouldn't know, but how I understand is that there are many reaction classes, I think on the order of 20, 30, but each of them has smaller diversity of building blocks. That's my understanding.
So basically you're trying by your models, you're trying those building blocks that I would say in the real database they wouldn't even consider.
Yes. That's my mental model. Yeah.
Okay. Thank you.
Yeah. Trading, like putting more risk on us, you could say. Okay. Thanks. So about another angle in which high-throughput experimentation is changing drug discovery. This is really recent. It's really interesting. Like we were to this conference Bio, which is top 1 conference for like kind of drug discovery, let's say. And well, last year there was nothing about what I will talk about now. This year this has been like really quite present. Like we can see a lot of trend in this. So this is quite novel in terms of, and we are enthusiastic about this in the company. So I just want to talk about this. It's not something we have as a big product yet, but maybe you will find this interesting. So here I actually talk about how usually hit discovery works when you order molecules from real. So you take real database. It's like, as I said, over 40 billion compounds these days. You maybe pick like 100 because you cannot pick that many more. Usually you pay like $30,000 per one order in hit discovery stage. Then they make them. It takes at least 3 weeks, often more actually to some extent, partially because of the customs, though that also depends. And then you do biological tests. So the whole process takes at least 6 weeks. I would say 6 weeks is not the median even. The median is probably larger. And this means kind of that you pay at least like $3,000 to $10,000 per heat, like actually more, but in chemistry, if you consider your heat rate to be between 1 and 10%, and the heat to be defined somehow such that this percentage is like that. And in some way, this revolution of direct-to-biology that I will talk about, I wouldn't say have saved the company, that would be an overstatement, but has been really, really useful. So we got a bit stuck. So SpaceM1, we believe, is a good product, differentiated, has good feedback. However, we are not at the moment ready to scale up operations. So synthesis from SpaceM1 has a physical footprint. And we don't want right now, for various reasons I won't go into, to have like hundreds of chemists just at the moment. So we had to start to reject, in a way, business or like direct our attention away from synthesis from SpaceM1. And the question was, okay, so do we just want to abandon SpaceM1? Do we want to scale it up, actually, and have this larger physical footprint? So we got a bit stuck at this difficulty, let's call it. So what makes a compound expensive? Well, purification, so that was mentioned, and use of materials, and use of labor, though that is not listed here. But that's what makes a compound expensive at this hit discovery stage. But what if we tested quote unquote directly? So it means without going through purification and test much less. So maybe I will just spend a second to explain purification. So let's say you have water and you dissolved in it salt, and you want the salt back, you know? That's the type of problem that chemists are facing. So you do reaction, and you want to extract things that are dissolved. And that's very difficult. It's very difficult, again, due to unpredictability, because there are different methods depending on what is the mixture. And furthermore, it's not that easy to predict accurately what method to use. And if chemistry is the hidden problem of drug discovery, then purification is the hidden problem of chemistry. It's really difficult. But what if we didn't do it? What then? So that's direct to biology. Usually when they make those libraries in the mode of direct to biology. Again, direct means you take the reaction mixture and dump it into your assay. You know, that's the idea. So you have some usually pipetting machine. Usually you make microliter at most of the solution, and you have like this whole maybe 384, maybe 96 plate of solutions. What is super exciting about this is that this workflow is really fast if done well. So that's a picture from Domainex. I probably do not pronounce the name correctly. A company that offers direct to biology based heat discovery or biological testing. So in 1 week, you can go from design to here, to screening results. It's ridiculous. It's really powerful in terms of timelines. It is like maybe 5, 6 faster times than normal. And I think that's important. I think time is something we underemphasize in drug discovery. We wait for results, we lose momentum, we lose time. So I think that actually matters a lot. Here's another one picture of that kind from Wuxi. Here they also add a step of resynthesis where you synthesize compounds that were active and then test them, which adds some time, but still can close in like 3 weeks, according, of course, to their materials. So if you were to compare direct to biology to typical hit discovery, you would have the following table. So you pay $10 maybe per compound, let's say, versus 300. You get, therefore, for the same number of for the same money, 30 times more compounds. Then you have to pay some extra fees, let's say, for example, for resynthesis. But bottom line is that you can pay for one hit like significantly less. So order of magnitude less. There is like 1 0 that's added. Sorry. So that's actually a game changer when it works. So does it work? Can you just dump a reaction mixture into your biological assay? Yes and no, and that's interesting. So basically, what research shows is that yes, you can, for some assays. And that's important because you can use some assay as a pre-filter to then test them in your assay that you are interested in. So from the paper, Nanoscale Synthesis and Affinity Ranking from 2018, well, the core challenge is false positives. Also, there is a problem of false negatives. So false positive is that when your assay lights up, but it shouldn't, right? False negative is when your assay lights up, but it should. Doesn't light up, but it should. Both are problematic. I would say false positives maybe are more important. One idea is to use affinity mass spectrometry. To maybe finish on time, I will describe the idea, maybe go a bit less into technical details of the paper, but you will get how it works. So again, the reason I'm talking also about this is that this is a beautiful example of rethinking drug discovery if you think about chemistry as the requirement. So we have direct to biology idea, 30 times cheaper hits, super fast timeline. How do we make biology work for that? Like, that's the question. Well, maybe use assay that work well with direct to biology. So one particular assay is affinity selection mass spectrometry. Not an expert in this area, but basically, you incubate your protein, if this is a target-based small molecule drug discovery, of course, and you incubate, some ligands bind, and then you separate the reaction mixture and measure with mass spectrometry the masses of different components of your solution. And basically the mass, the core idea is that the mass tells you which ligand has bounded if ligands have different masses. So the readout is peaks, right? And the highest peak, let's say, or like, and from the peak masses, you can say which ligands have bound. So from affinity mass spectrometry, you can get yes, no. Now, there are techniques to get a more quantitative answer by changing the concentration of the protein so that ligands that are less competitive go beyond detection threshold. That's also an interesting technique. But main thing to remember is that you basically measure the mass of the ligand protein complex. And the reason it works with just dumping the whole reaction mixture, kind of, and why many papers talk about this, is that now you just, these different components of the reaction mixture will bind to the protein, but your assay won't light up because you measure precisely the mass. And only your ligand has the mass that you, only the product you care about has the mass. Mostly, like there are maybe some edge cases. Yeah, so let me finish this section here. There is not that much later, but I don't want to overrun with time. So to summarize, it was, I think, a tour of the topic, so I thought a summary at the end would be nice. So organic chemistry is unpredictable for humans. So basically, if you remember this from the talk, I would be super happy. So chemistry is unpredictable for humans, which results in slower and more expensive drug discovery. And there is a revolution in high-throughput experimentation. So we both generate the data, we have ideas to also synthesize molecules in high throughput, which basically changes the game of the drug discovery, where you can make molecules cheaper, more diverse, and in different ways. In particular, Molecule 1 has been at the forefront of some aspects of this, this trend. So we have generated probably the largest of its kind microliter scale reaction dataset, used it to build superhuman models, and shown that we can offer a space of molecules that has differentiated diversity. And we believe, in general, the high-throughput experimentation will revolutionize drug discovery by opening ways to make novel molecules that are not now made because we reject them as too complex, and also cutting dramatically the cost of one drug approved, because we'll cut down both manufacturing cost and discovery cost. This talk was more about discovery. Yeah, I mentioned what we did at Molecule 1, actually. And maybe one closing thought. What I am personally most excited about, I think, is how this trend of high-throughput experimentation connects to the recent advances on AI scientists. I think in general, well, I think we all agree in general, or like domain agrees maybe, that building an AI scientist is a very exciting and to a large degree attainable goal, and that's what many big labs are now focusing on. And in particular, I think an AI chemist is necessary to solve chemistry. So by solve chemistry, I mean you can get any molecule you want as long as it is like physically reasonable. And I think this will really transform drug discovery, I think. And for that AI scientist, high-throughput data set will be necessary as a tool for it to explore chemistry. It cannot make reactions one at a time if you want to solve chemistry. So one summary of the whole talk is large-scale experimentation chemistry will revolutionize drug discovery, and we can already use it in novel products such as direct-to-biology and novel types of molecular spaces. So that's all. Thank you very much for your attention in the morning. Yeah. Any questions? Yeah.
Well, if there are no questions yet from the audience, I have one. Since you've finished, sorry, did you have a question? Apologies.
Hello, thanks for the amazing talk. I also love the Breaking Bad references. It's my favorite TV show. It was amazing to see here. I have a question about the synthesis model that you developed. You said you're using conformers, but then the model has to correct them automatically inside the box, let's say.
I mentioned specifically that I think it learns to do that. I'm not sure if it does for sure, but that would be my guess.
But then my question would be, do you then even need to use the 3D information? Or can you switch to a 2D or 1D representation to predict that?
Yeah, it's probably possible, yes. We would need to retrain the pre-trained backbone to work on that, or maybe just train with that. So in short, probably yes. Probably if I were to change those molecules to, let's call them pancakes, like no conformation, probably it would work similarly. That's my guess, but we haven't tried that.
And my second question would be actually about the transfer learning part, because you said you pre-train and then I guess fine-tune. Could you elaborate on what you pre-train on and what you fine-tune with?
Of course, of course. So that's a set of ways available to everyone from Unimol V2 specifically. By the way, the whole family of Unimodels is amazing, and I really recommend their research. In particular, this model was trained on a few tasks. One of them in particular was, if I remember correctly, predicting the conformation, denoising the conformation. Might be wrong, I don't recall precisely, but that was what I think one of the tasks. Yeah, I'm not sure what other actually. So sorry, we'll have to go back to the paper to see that. Yeah.
I feel like kind of monopolizing the microphone. Okay, I wanted to ask maybe in the end, your general view for the company for the future, because as far as I understood right now, so you're having this pretty accurate models for reaction prediction, and you're kind of providing for the clients the space M1, which is like a space of also synthesizable compounds, which you're actually doing probably somewhere in the labs. And you're right now, as far as I understood, switching to the direct-to-biology also. So that's would be your like a next step in the company, like a product or
potentially. We are experimenting with that right now, and also our software is really an important offering for us. So it's not only laboratory.
Okay, so it's both retrosynthesis
and
space M1.
Yes, and I think in the end they connect, because in the end the goal is to build like really highly accurate prediction software. So.
Okay, thank you.
Thank you.
One question I wanted to ask is towards those final thoughts on AI scientist. Where in your sort of scope would that fit in, and whether you are already, I don't know, evaluating any of the existing systems in terms of how well they might perform whatever you're seeking to do with that?
Right. It's interesting, and you can also see that in other companies, is that in some weird way, the lack of automation is also a bottleneck to AI scientists in biological sciences. For example, there was a very interesting paper by Future House where they claim that AI scientist has invented an important molecule from biological standpoint. However, all experiments were done manually by humans directed by AI. I think that's interesting. So the way we think about this is that as we advance more, the high throughput laboratory, there will be more and more room. But right now, to come back to question about random exploration, we think that there is a lot of low-hanging fruits in taking on a problem and kind of randomly exploring. And maybe after we do it for a while, then there will be a lot of room for more kind of AI scientist-like applications. So right now, we focus a lot on building these products. We have less direct focus on building AI scientists. But as we transition, we'll definitely explore that, and kind of that's definitely something very exciting for everyone at the company.
Thank you.
Thank you for your talk. I was curious if you have any more insights on, you had this little graph for scaling and the amount of data and the performance of the models.
Right.
And it was going from, I think, 10,000 to 50,000.
Yeah.
And it seemed like on the transformer model, the trend was almost like linear.
Yeah.
So is your intuition that if you just keep, if you 10x the data or 100x the data, you'll essentially see these amazing gains in your model? Is the scale here the ultimate goal? Or?
Yes, but maybe not within one reaction class. But for example, now we have a very exciting project where there is a very, very promising reaction that someone has just published, but no one is using that in drug discovery. So we took that and we want to generate 50,000 data points around it as the first in the world and see whether it works. So it's that type of work that I think now is the biggest opportunity for us. After that, I think as we explore that, and we can do that in parallel, there are very exciting things around trying to understand more deeply chemistry. So trying to be more like scientists about chemistry, and then those things can have very big impact, for example, on manufacturing. So definitely big opportunities, but for now, we focus a lot on this high throughput experimentation to kind of build our kind of niche, let's say, there.
Thank you.
Yeah.
All right.
Marvin?
Sorry. It's really hard to scan the
Yeah, thanks. So how transferable is the high throughput experimentation data to then kind of higher
scale?
Yeah, higher scale.
Right. So we haven't tested we take high throughput experiments and move them to synthesize 100 kilos of a compound. We haven't done that. I don't know. But what we do routinely is we take synthesis in this microliter and move it to more grams or higher milligrams, and we see pretty good correlations. We actually optimize the high throughput experimentation protocols to achieve their more correlation, because there are aspects of high throughput experimentation which do not transfer well, and we try to avoid them.
All right. Could I ask you for one more round of applause for our speaker?
Thank you. Thank you.